Hello everyone, you are so much welcome at Medical Sciences by Naftali Muhumza. As we continue with HEXOSE monophosphate shunt, or what we can call pentose pathway. In the previous video, in part 1, we have looked at the process and the regulation of HMP pathway. And in this video, we want to look at the significance and the clinical aspect. How can we apply this pathway into our clinical setup? And as we always remind you, subscribe if you haven't subscribed, like the video and share your comments. So we want to look at how is HMP pathway, or what you call pentose phosphate pathway, how is it significant in our medical setting? And we know that this HMP pathway, the way we saw previously, it is divided into two processes, which is oxidative phase, it has oxidative phase and non-oxidative phase. And in the oxidative phase, we saw production of 6-deep-H. We saw production of 3 carbon dioxide molecules. We saw production of ribulose 5-phosphate, which was acting as a precursor of the non-oxidative. And under non-oxidative, we looked at production of ribose 5-phosphate. We saw production of fructose 6-phosphate and glyceroldehyde 3-phosphate. So these were the major products produced during HMP pathway, whereby we are producing 6-NADPH, 3 molecules of carbon dioxide, ribulose 5-phosphate, which act as a precursor for the non-oxidative phase, leading to production of fructose 6-phosphate and galactose and the glyceroldehyde 3-phosphate together with the ribose. And we're going to see the significance of all these products in our body metabolism or body system, whereby we are going to see that significance of this pathway. Among the significances is production of ribose 5-phosphate, and this ribose 5-phosphate is very important in the synthesis of nucleic acids. Synthesis of nucleic acids like RNA and DNA. Because we know that RNA has ribose, the pentose sugar is ribose, whereas DNA is deoxyribose, synthesized from ribose, which is an intermediate of HEXOSE monophosphate shunt. And another significance, number 2, is that it enables interconversion of pentoses to hexoses. Conversion of 5 carbon to 6 carbon occurs during the non-oxidative phase of the HMP pathway, whereby we saw transketolase, or by the process of transketolation together with transaldoylation. Then another significance, we shall see production of 3 carbon dioxide molecules, which plants use for photosynthesis to synthesize their food. And another significance of this pathway, we are seeing production of fructose 6-phosphate and glyceroldehyde, fructose 6-phosphate and glyceroldehyde 3-phosphate, and these guys are used by the body and they undergo gluconeogenesis, whereby they undergo gluconeogenesis to produce glucose, and this glucose can be recycled in the HMP pathway. So production of these fructose 6 and glyceroldehyde 3-phosphate undergo gluconeogenesis to produce glucose, which can be utilized 10% in HMP, and another one goes into glycolysis and glycogenesis. As we move on, we have seen the importance of these. Now we want to go to the importance of NADP. We saw production of 6-NADPH, and this reduced NADPH is very responsible in many metabolic processes and one of them is NADP. It enables detoxification of drugs by microsomal cytochrome P450 isoenzymes, which are responsible for detoxification of drugs. They utilize NADP-H. We saw this NADP-H also playing a big role in biosynthesis of steroid hormones. We saw it very important in biosynthesis of neurotransmitters, in the biosynthesis of cholesterol, also in biosynthesis of cholesterol. We see it in the de novo synthesis. We see it in the de novo synthesis of fatty acids, de novo fatty acid synthesis. So these are the processes that are supported by NADP-H, including uronic pathway, also utilizes NADP-H. So these processes utilize NADP-H. So synthesis of fatty acid, synthesis of cholesterol, synthesis of neurotransmitters, steroid hormones, all of these ones utilize NADP-H as a precursor. So we see NADP-H being a proton donor in these processes. In these processes, it acts as a proton donor. It acts as a proton donor in these processes. So these are the major functions of NADP-H in our body. And this is the significance whereby we have seen significance 1, 2, 3, 4, then 5 into different processes. So this one you can break it even into more 5 points. Don't always combine it. So we have seen the significance is majorly synthesis of DNA and RNA by ribose 5-phosphate, interconversion of pentoses into hexoses occur in this process. Plants utilizing carbon dioxide produced to carry out photosynthesis. And we have also seen fructose 6-phosphate and glyceraldehyde, which are the products utilized under gluconeogenesis to produce glucose. And we have seen also the NADP helping in the detoxification of compounds. We have seen acting in the biosynthesis of steroid hormones, neurotransmitters, de novo fatty acid synthesis, synthesis of cholesterol, and in uronic pathway. And the last we can add is that it also plays a role in phagocytosis. Whereby it produces superoxides. And these superoxides are the ones that kill the infected cells. And lastly among the significance is that this one plays also a big role in it reduces oxidized glutathione. This oxidized glutathione to reduced glutathione. And this glutathione, if oxidized glutathione accumulates, that if we have oxidized glutathione, this is oxidized glutathione is reduced glutathione by an enzyme known as glutathione reductase, and this enzyme utilizes NADP-H, forming NADP+. And this one, the reduced glutathione which is formed, this is reduced glutathione, is responsible for removing hydrogen peroxide in the cells. That whenever there is accumulation of hydrogen peroxide, which can result into formation of superoxides which are oxidant, which can oxidize many components, especially red cells, leading to hemolytic anemia. It is broken down by this reduced glutathione. In presence of glutathione peroxidase, and whenever this glutathione peroxidase converts this, we form water and oxidized glutathione again. And this is the protective role of this pathway, which supplies NADPH, which prevents accumulation of oxidized glutathione, hence preventing even oxidation of hemoglobin to methemoglobin. And when hemoglobin remains stable, red cells are protected by this process. So our red cells do not suffer stress due to presence of NADPH, which is responsible for reducing oxidized glutathione. So this is the point number last we can talk about. 5, 6, and 7, but we have seen number 5 has many points. So after seeing on the significance of this pathway, the last part we can look at is the clinical aspect. And look at the clinical aspect of this pathway of HMP, or what we call pentose phosphate pathway, or HEXOSE monophosphate shunt. And this shunt plays a major role, whereby there are major two clinical aspects, whereby there are people who have glucose 6-phosphate dehydrogenase deficiency, and we have another condition which we call favism, and the last condition shall be Wernicke-Korsakoff's syndrome. So these are the major three clinical aspects of, in any way, whenever there is when HMP pathway is hampered or is obstructed or is inhibited, we can see these conditions coming into play. And this glucose 6-phosphate dehydrogenase deficiency, individuals with this is a mutation, is due to mutation whereby it is acquired via X-linked gene, is acquired via X-linked, and this clinical presentation of G6PD, after being acquired as X-linked, we see it whenever there is a deficiency of this enzyme, there is accumulation of oxidized glutathione. This is oxidized glutathione accumulates in cells. We saw the importance of G6PD was being used to produce NADPH, which NADPH reduces oxidized glutathione to reduced glutathione. So in case there is no G6PD, means there is no NADPH, and no NADPH, oxidized glutathione accumulates, and when it accumulates, it increases red cell hemolysis, RBC hemolysis. How we see this one happening? It happens whereby in case in red cells we have HB having ferrous iron, which is able to carry oxygen. It is oxidized by accumulation of oxidized glutathione to form methemoglobin. And this methemoglobin has iron in ferric state, and whenever hemoglobin has iron in ferric state or in iron 3 state, it does not carry, it fails to carry oxygen, respiring cells. And we are seeing that this increased hemolysis, so this one results into hemolytic anemia. This one results into these people with G6PD suffer from hemolytic anemias. And these hemolytic anemias come when they take oxidant drugs, when they are given oxidant drugs or drugs that cause oxidation, like antimalarials, antimalarials, malarials like primaquine. Whenever someone is on primaquine and is 6-GPD deficiency, someone on aspirin and sulfonamides, they tend to suffer hemolytic anemias. So this one comes when they are exposed to oxidant drugs like antimalarials such as primaquine, dapsone, aspirin, which leads to hemolytic anemia. And these people with G6PD deficiency, another good part of them is that they are not prone, or they are not prone to malaria. Because we see even malaria parasites, they have their obligatory life cycle in red cells. So whenever there is increased destruction of red cells, even the malaria parasite does not finish its life cycle. So you find people with G6PD deficiency rarely suffering from malaria due to the accumulation of the methemoglobin, which destroys the RBC before malaria finishes its life cycle. So they are not prone to malaria. They are not. Or they don't easily suffer from malaria. And this was the first one. The third one is the favism. And this favism, here there are people whereby whenever they take fava beans, these fava beans in Greek you can call them vicia fava, that when they take them uncooked or undercooked, or they inhale the pollen grains of this fava bean, the fava bean taken inhibits G6PD. So the fava bean inhibits G6PD enzyme, and after inhibiting it, there is no production of NADP H, and without no production of NADP H results into accumulation of oxidized glutathione, leading to hemolytic anemia. So these people, favism, they also suffer from hemolytic anemia due to the component of the fava bean which is got by taking uncooked beans or undercooked, or the pollen grains from the plant of the fava bean, inhibiting G6PD dehydrogenase enzyme, leading to low levels of NADP-H. And without NADP-H, glutathione, which is oxidized, accumulates in the cells, leading to hemolytic anemia. Which hemolytic anemia, there is increased destruction of red cells. Then lastly is this was the second, this is the third, which is the last one, is the Wernicke-Korsakoff's syndrome. And in this syndrome, we see that people have they have genetic variant in the transcatalase enzyme. And whenever there is a variation in transcatalase, it fails to bind to thiamine pyrophosphate. And whenever there is no binding of thiamine pyrophosphate, it leads to different neuropsychiatric disorders, neuropsychiatric syndromes like dementia, like memory loss. They suffer memory loss, they suffer paralysis of eye movement, they suffer from mental confusion, any psychiatric problem, depression, you can mention them. So that is what we call Wernicke-Korsakoff's, due to variation in transcatalase enzyme failing to bind to thiamine pyrophosphate, which is able to transfer the 2-carbon sugar from keto sugar to aldose sugar. So whenever that one happens, we see accumulation of these ketoses in the of aldoses in the brain, leading to neuropsychiatric problems like memory loss, there is a depression, there is paralysis of eye movement, and mental confusion, and sometimes it can result into death. That is what we call Wernicke-Korsakoff's syndrome. So the clinical application of this HMP is seen in G6PD hydrogenase deficiency, seen in favism whenever people are prone to fava beans, and also variation in transketolase leading to Wernicke-Korsakoff syndrome, which is a neuropsychiatry problem. Thank you so much for listening. Always reminded to subscribe, share the videos, and like the video so that we can continue to grow. Thank you so much. May God bless you.